Delving further into the structure of an antibody, what it looks like, that sort of thing. An antibody has a constant region, as you can see here, composed of a heavy and a light chain, which stays the same for every antibody. It just kind of stabilizes the protein as a whole. There are a variable region of the antibody, and this is the region that binds to the antigen. That's the one that changes all the time. And then there is a crystallizable fragment region. This is the part where other antibodies, leukocytes, the complement system can bind to exert its effects. So once again, just a Y-shaped protein that binds to the antigens with the variable region, and then other things can come bind here to exert their effects. In particular, the way that antibodies are able to assist in killing pathogens are all of these things you see here. So precipitation, it can bind to the pathogen and make them soluble. So oftentimes pathogens are present in tissues, and by making them soluble, they'll be able to precipitate out of the tissues and be in the bloodstream, basically. And it's easier to eliminate, easier to interact with more cells when they're in that capacity. Agglutination is also something that it is responsible for. So this is where once the antibodies have bound to a particular pathogen, the crystallizable fragments will bind to other antibodies, which will kind of clump all of the cells together. And, if all of these pathogens are clumped together, they're easier to eliminate. The other way is neutralization. So the antibodies will bind to a protein or a receptor that is dangerous, meaning it's like responsible for killing cells in the body in some capacity. So it's basically like putting a sheath over the knives of these dangerous bacteria. They can't hurt the body's cells anymore. The other thing they do is they trigger release of histamine, which also kind of just targets other cells to come in and help out. And last but not least, they are able to signal a system called the complement system, which acts really similar to natural killer cells or cytotoxic T cells, in that it's a system that once it binds to the antibodies, releases perforins to make holes and granzymes to chop up parts of the pathogen and ultimately kill it. And then as you can see, all of these other four functions are part of a process called opsonization and phagocytosis. So opsonization is just the process of basically marking something for destruction. So that's what they're doing in these various things. And then once they're marked for destruction, they go through phagocytosis. It's just a fancy word for one cell eating another cell. So the pathogen is engulfed by some kind of macrophage or other phagocyte. There are different types of immunoglobulins. The different immunoglobulins are separated because they have different crystallizable fragment regions. So those regions that can bind to different proteins, different leukocytes, that sort of thing. I'm not going to go in particular to a lot of these. I guess the main ones are immunoglobulin E, that are usually activated by allergens, and then they will activate mast cells and basophils. And immunoglobulin G is the main one you'll see in immune system defense. Last, connecting immunoglobulin and B cells all the way here, we have a term called somatic hypermutation, which is just a fancy phrase to describe how B cells are able to create antibodies for particular antigens. They have such a high mutation rate in the genes that are creating these variable orange regions of the antibody that binds to the antigen. They're at such high mutation rates, and they just produce a bunch of different variable regions initially, and then whichever form of that variable region matches up and binds to the antigen most effectively, that's the one that will end up receiving growth factors and will end up surviving. And then the other ones, because they don't receive growth factor signals, they'll end up dying out. So it's kind of the idea of evolution or survival of the fittest, where the antibody that is produced with the highest affinity for a given antigen is the one that will survive, and all the other ones will die out because they don't get those growth signals. Once they have actually produced an antibody for a particular antigen, it can switch between those different classes of immunoglobulins so that it can kind of cover a wide array of other cells that it can bind to and functions that can happen to it. So if it switches to immunoglobulin M, it might be used in agglutination, whereas if it uses immunoglobulin G, it might be used in neutralization, things like that. So that's why class switching is important.